I'm Jackie Borg. I'm a researcher at the Karolinska Institute, and I also work as a clinical neuropsychologist in Stockholm.
I am Gina Griffioen. I'm a PhD student of Jackie, and besides researcher, I'm also a psychiatrist working with people with intellectual disabilities and severe behavioral problems. And I also have a sister with neurofibromatosis, which has a lot of features in common with autism.
The purpose of this video is to give an introduction to our work in PET imaging in autism and autism-related traits. PET is an acronym for Positron Emission Tomography, which is a brain imaging method to study proteins in the brain. I'm very aware that language and terminology is important for many people. So I thought I'd start with explaining the terminology that I will use myself. We know that many autistic people prefer to be referred to as autistic, but many other people also prefer to be referred to as having autism rather than being autistic. In our consortium, I have followed the discussions about language, and I've understood that many or most maybe of you AREPs, or most of our AREPs prefer to be referred to as autistic. However, in my work as a clinician, I have actually found that a minority of my own patients in the healthcare system wants to be referred to as autistic. On the contrary, when I've asked around, the vast majority prefer to use the expression, I have autism, or I have a diagnosis of autism. And now I should of course mention that my patients, they are patients because I work in the public healthcare system. So they will actively seek me out if they need or want psychiatric treatment because I work in psychiatry. So that is of course a different sample compared to our AREPs. I do, however, feel that if I only use the word autism or autistic people when I speak to you in the consortium, then I feel that I'm sort of going behind the back of my own patients and letting them down because I know they would not like me to talk about them as autistic people behind their back. So because there is no solution where everyone is happy about the language we use, I have actually chosen to alter. So sometimes I will say autistic people, and sometimes I will say people with autism.
Yeah, I think I agree with Jackie's view. What I perceive from my patients, and I'm also working in psychiatry, that they also would like to be called people with autism, and even my sister. But I think it's different for everyone, and that's fine. But it makes it very hard for us to do it right, so to say. So I'm following on also as Jackie does, like alternating and hoping not to be too harmful or harmful, yeah, to offend anyone with that.
We also know that there are differences in language and culture. And for example, in Sweden, we actually used to say the formal word was autism spectrum condition in Swedish until a couple of years ago, when we in Sweden started to use the words autism spectrum disorder in order to be better aligned with the English word, because the diagnostic manuals are in English. But many people in Sweden still say autism spectrum condition, because that used to be the formal word in Swedish for a very long time. PET doesn't measure activity in the brain. It measures markers for neurochemical signaling. And in our brain, we have about 80 to 100 million brain cells or neurons, and they communicate with each other all the time. That's how the brain works. And what we can do with PET is to examine those proteins that are that receive or send these chemical signals. When we talk about targets, we actually mean specific proteins in the brain, and those proteins can have different functions. What we study with PET is mostly a kind of protein target that is called a receptor. And receptor means it actually receives the neurochemical signal from another neuron. So that's why it's called the receptor. So very often we could actually say receptor instead of target, but not all proteins we study are actually receptors. And many of the targets Gina and I are going to talk about today are not receptors. It's transporter proteins that have a different function. But we also say targets because these proteins are actually the targets for almost all psychotropic medicines that we know of today that are used in mental health care, in psychiatry, and also some in neurology.
A psychotropic drug is a medicine which is affecting brain function, and with that, hopefully psychiatric symptoms or things like attention or other brain functions. So first, we need to have a tracer which binds to the molecule or protein of interest or the target, like Jackie explained. And then we need to label it with a radioactive particle. And that sounds a bit scary. However, it's not that bad. Radioactivity is all around us. It's from radiation comes from the Earth or from the universe. And actually, when flying in an airplane, you have more radiation than being on the ground. And the doses used in our studies are comparable to a flight from, let's say, Stockholm to New York. And those doses are not related to any health risks. So the person receives an injection with a tracer, and the tracer then travels through the blood and ends up in the brain, where the tracer will bind to the target or the protein of interest. Then this tracer sends out its radioactivity, and our PET system will be able to detect that. And then with complicated computer techniques, we can then calculate where the tracer is and the amount of protein in the brain and where it is located.
Usually PET images are shown in a color scheme called the Sokolov scheme. And in that scheme, red is the hottest, so that's the highest concentration of the protein. And blue is the coldest, so that means you have almost no protein there at all. And black means it's absolutely zero, you have nothing there at all. So PET can be used to study the neurochemistry in a certain diagnosis compared to another diagnosis or people with no diagnosis at all. But it can also be used to study the general neurochemistry behind specific human behavior. For example, if you have a low or high amount of dopamine D2 receptor, or you have a high or low amount of serotonin 5-HT1A receptor, this actually makes a difference, regardless if you're a person with a diagnosis or a control subject. With PET research, we have found previously that differences in brain protein amounts actually correlates to various cognitive functions, personality traits, and other behavioral functions. And that's why we think it's so interesting to study, because it tells us something about the work, the brain's work behind human behavior?
So we study trait and behavioral measures in people with and without diagnosis in order to learn more about the neurobiology of certain traits. And it can be autistic traits, but it can also be cognitive traits like attention or how good you are at certain tasks. Don't just only study autistic people versus neurotypical people, but we also want to know in what way are the differences, but where is it? It's not so that it doesn't rely on the diagnosis. We also know that like all the traits are normally distributed in society or in people. So some might have a lot of the traits, some might have very little. We want to see if that correlates to certain measures we do with PET. So in order to understand what's happening.
In addition, we can also use PET as a tool in drug development because when we can associate a certain behavioral function with a certain brain function, it means we can also aid drug development by with the information that if a certain protein is associated with, let's say, a cognitive function, if you want to change this cognitive function somehow, let's say you want to improve a memory function, then you can try to make a drug compound that actually targets the protein that we found was related to memory function. And it also works the other way around. So let's say you don't want people to have lower cognitive function when they take this medicine. Then we can aid drug development with the information that avoid this certain target because it is related to lower memory function.
In regard of autism, where we know the spectrum is so diverse, we'd actually like to discover certain behaviors or traits or features and how they correlate to certain brain chemistry functions.
What we're aiming at here is to be able to develop drugs that specifically target a certain behavior that some people may want to have treatment for, whereas we want to leave other autistic traits unchanged. So the idea is that you could develop drugs that are very, very precise. Drug development should only be for people who actually actively seek help or treatment for certain aspects of their behavior or thinking patterns. Sensory sensitivity is actually a good example because it is something that many autistic people acknowledge they experience difficulties with. So if a person with autism experiences a certain problem, for example, with high sensitivity to sensory stimuli, what we would like to do with PET is to aid drug development that can help finding a drug that would target specifically this sensory sensitivity and not all the other autistic traits that may be part of that person's adaptive abilities and well-being.
So another example can be difficulties of being able to read people's facial expressions, where we specifically look at in our studies, because that can take a lot of energy from someone. And if we know the mechanisms behind this and maybe can find a drug targeting this, so it will be easier and less energy consuming, that will also aid someone who's having troubles with it. In the AIMS-2-TRIALS projects, we focus on several systems, but this webinar will mostly be about the serotonin system. And I would like to tell you something about that. So serotonin is a neurotransmitter what Jackie also already explained, where one nerve cell is sending out information to the next nerve cell. It's using chemical messengers, and serotonin is one of these messages. But you have more messengers like dopamine, glutamate, GABA. These are also messengers. Serotonin is important in many regulatory brain and body functions, like mood regulation, body temperature, appetite, sex drive, what do we have now, social behavior. That's also maybe a lot of antidepressants also act on the serotonin system, and a lot of side effects are related to this. So more appetite or loss of sex drive. Another important function of serotonin is brain development. It guides the formation and guiding of neurons during brain development. And we have reasons to believe that this is already might be impacted in part of people with autism or autistic people. We know, for example, that in 1/3 of the autistic people, there are high blood serotonin levels. But we also know from post-mortem studies, these are studies where we investigate brain tissue from deceased people, that we found higher levels of serotonin system there. One of the things we're going to talk about in the webinar is also what we found with PET in the serotonin system, but then in living people.
So you can study the serotonin in a less precise way by studying serotonin levels in blood or in cerebrospinal fluids or something like that, which is called peripheral measurements because it's not in the brain. But by doing that, you actually study serotonin levels in the whole body. And because serotonin, the brain cannot use serotonin from the body. It has to make its own serotonin because serotonin doesn't cross the brain-blood barrier. So you have to use the tryptophan from your food in order for your brain cells, your neurons, to synthesize serotonin, which means that studying serotonin in the body, not in the brain, it may be a flawed method because it maybe doesn't say anything at all about the serotonin in your brain.
So we are able to study the serotonin system more precisely in living people and compare autistic people with neurotypical people, but in relation to the serotonin system or some aspects of it.